Hello everybody. Have you ever wondered why so many Europeans have blonde hair? Well, today, along with 3 of my friends, we'll inform you on the genetically small difference that determines this hair color. What if I told you that switching just one letter in your DNA could change a brunette to a blonde? What? How? Well, in a little bit more scientific terms, researchers have discovered that just one nucleotide switch from A to G results in the blonde phenotype. They first used fish, then mice, to see if they had the same effect, and as a result, they discovered that the blonde switch changes the activity of the gene called the KIT ligand.
This graph shows SNPs on chromosome 12. SNPs are single nucleotide polymorphisms, which means a single nucleotide was changed. The RS128212556 SNP is the location of the AG switch most highly associated with blonde hair. This picture shows the difference in fur color of mice with and without the SL-PAN mutation. The SL-PAN mutation is a mutation of transcription factors on chromosome 10 in mice, which produces fur color. This gene shows incomplete dominance since there is an intermediate phenotype.
This graph shows the genetic basis for hair pigmentation in various mammals, which shows increased activity at the RS128 site, relating this area to blonde hair. This confirmed the suspicions of our researchers, who believed this area of genetic information would be key to discovering the origins of blonde hair. The KITL gene is located on the long arm of chromosome 12 at position 22. More precisely, the KITLG gene spans roughly 10,000 base pairs on the chromosome. This area of the chromosome also encodes for eye pigmentation, which led researchers to believe that the two traits may have been correlated.
Here we have the LAC-Z expression for developing embryos, the ancestral allele on the left, the blonde allele in the middle, and the deletion of the RS128 position on the right. The kidney provided a control in this experiment. Remember that the LAC-Z expression we are interested in was visible there as well as the hair follicles. The LAC-Z activity was observed in all the developing embryos, but there was no repetitive difference noted between the A and B embryos.
Now we're looking at LEF1. LEF1 is a transcription factor and is known to be expressed in hair follicle development. Studies have shown that mice with LEF1 knockout appear lighter in color due to the lack of production of melanin. The characteristics of LEF1 prove it to be the best transcription factor to act on KITLG. The blonde-associated allele in humans is the change in nucleotide from adenine to guanine within the LEF binding site. The base pair change causes a reduction in the responsiveness of LEF1, which in turn causes hair to appear lighter in color. Here there are 3 types of mice that were used to study the effects of the KITLG expression. The mouse on the left was used as the control. It had the deletion of the RS site. The mouse in the middle was carrying the allele associated with blonde hair, and as you can see, it is noticeably lighter pigmentation than the mouse on the right that has the ancestral allele. Now let's wrap things up. Through this article, we've learned that the current research shows us the molecular basis of common human hair color phenotypes. Although it is a highly noticeable trait in humans, until now, people have had little understanding of it. The study of the molecular basis for blonde hair has shown us the impact of a tiny change in DNA. Thank you for watching.